Hello, welcome to this IUIS Frontiers webinar in the series on COVID-19. We're just going to wait another 30 seconds or so for people to be joining us around the world. This is very exciting to be talking to people from all over. It's fantastic. So I'm in Wales and our two presenters today are in England. So we're actually in different countries, but part of the United Kingdom. So just a couple of seconds. So I'll just start by introducing myself very briefly. My name is Ann Ager. I'm Professor of Cellular Immunity and Immunotherapy at Cardiff University. My interest is in T lymphocytes and T lymphocyte trafficking. And it's a really great honor for me today to introduce two new, young, highly talented fellows who've just literally started to set up their own laboratories, both at the University of Manchester in the Lydia Becker Institute for Inflammation. So without making priorities, because I don't want to do that, I'm going to start with Elizabeth Mann, Lizzie, who I actually do know. So Lizzie's background, she did her PhD not so long ago now with Stella Knight on dendritic cells, and really has moved from Baltimore and Glasgow and now Manchester, focusing on macrophages and how they are tailored to their mucosal environment. And so Lizzie is really a macrophage person. And then Madhvi Menon, I want to introduce. So Madhvi is really, I think, just arrived in Manchester just before the lockdown. So was really thrown in at the deep end with this pandemic and COVID-19. So Madhvi is a B-cell immunologist and has worked very much on autoimmunity. So this complement of these two different very important immune cell types in responses to this new virus is really important to understand. And without further ado, all I want to say is that I think I can say from Lizzie and Madhvi that they have worked very, very closely together in a very short period of time. And what's great is you've met each other very recently and you've become good friends through this fantastic collaboration. So without further ado, I'm going to hand over. Madhvi's going to open the presentation on longitudinal immune profiling, revealing distinct features of COVID-19 pathogenesis. So welcome to the world, and you have this fantastic opportunity to hear it from the people who've been doing the frontline work. So over to you, Madhvi.
Thank you, Anne. Can we have this slides up, please? Okay, so first of all, I'd like to thank the organizers on behalf of Lizzie and myself for the invitation and this fantastic opportunity to present our research to you today. So Lizzie and I will be talking about our work on the longitudinal profiling of COVID-19 patients and discuss some of the distinct features that we've found to be associated with disease pathogenesis. The work we're presenting here today is the result of an enormous collaborative effort between immunologists at the University of Manchester, along with clinicians, research nurses, and clinical coordinators at 4 different hospital sites in Greater Manchester. Our study is called the CIRCO study, or the Coronavirus Immune Response and Clinical Outcomes Study, that is being led by Professor Tracy Hussel, along with 5 other colleagues at the Lydia Becker Institute of Immunology and Inflammation at Manchester. And the idea really was to bring together our different immunology expertise to work as a team in improving our understanding of the immune dysregulation in COVID-19. So for our study, we collected blood samples from patients longitudinally. So at serial time points starting from the time of hospital admission all the way until discharge, typically every 1 to 2 days. The blood samples we collected were used to measure levels of cytokines and chemokines in the serum of the patient, as well as to phenotype the different immune cell subsets using flow cytometry. In addition, we're currently in the process of sequencing the whole blood samples from our patient cohort to study the transcriptome and the B and T cell repertoire in COVID-19 pathogens. I should mention that majority of this, actually the entirety of this work was done real time with fresh samples, which gave us the unique possibility to be able to study populations of neutrophils, which otherwise wouldn't be possible with frozen banked PBMCs. So within our patient cohort, COVID-19 patients were classified as either mild, moderate, or severe disease, based on the supplemental oxygen levels required to maintain their target oxygen saturations. So mild patients typically required less than 3 liters of oxygen, whereas moderate patients required between 3 and 10 liters. Severe patients, on the other hand, required more than 10 liters of oxygen, were managed in ICU, and some of them went on to then requiring ventilator support. For our study, so far we've analyzed a total of 73 patients cross-sectionally, 49 patients longitudinally, and compared their immune phenotypes to 13 healthy controls. Due to the difficulty in recruiting healthy controls at the height of the pandemic and the lockdown, majority of our volunteers are in fact healthcare workers and university staff. When we first started this study in early April, there was very little known about the immune response to SARS-CoV-2 infection and the clinical management of disease. So there were really 3 key questions we were interested to address over the course of the study. The first being, how does the immune response associate with the severity of COVID-19? Second, can we identify biomarkers being specific features of the immune response at hospital admission that can predict eventual development of severe COVID-19 symptoms? And third, what are the immunologic parameters that track with patient recovery? So to do this, we used flow cytometry to identify distinct immune cell populations and their activation states at hospital admission, and analyze this data primarily via flow cytometry. So in the UMAP plot shown here, the UMAP plot shown here is representative of a healthy individual, where you can see all the distinct immune cell subsets, the granulocytes, monocytes, NK cells, B cells, and T cells. For our study, we primarily focused on the cells highlighted in red, so namely the neutrophils, monocytes, T cells, and B cells, which will be the focus of our talk here today. When we compared the immune cell subsets between the healthy individuals and patients with COVID-19, we saw that whereas patients with mild disease had an immune profile similar to that of healthy controls, patients with moderate and severe disease displayed an expansion of neutrophils here in orange and a reduction in majority of the other peripheral immune cell subsets. When we look at this summary data, we see that there is in fact a significant reduction in the frequency of total T cells and B cells in the periphery at hospital admission in patients with severe COVID-19, and that this is mirrored by a significant expansion of CD16 positive neutrophils in these patients. So in these graphs, the severe patients are represented as black circles, moderates in blue, the mild in green, and the healthy controls in white. When we then looked at other immune cell subsets, we found that monocytes, NK cells, and eosinophils, although showed some variability, were not dramatically impacted. However, basophils and plasmacytoid dendritic cells were also decreased in patients with severe disease. Next, we wanted to track these immune cell subsets over the course of disease and in order to assess how the kinetics of the immune response and how this associates with the clinical outcome. So here we have the frequency of T cells and neutrophils plotted against the days of reported symptoms in moderate patients with the different colors highlighting individual patients. So for this analysis, we typically have patients from the time of hospital admission until discharge. And what we can clearly see is that over a period of time, the majority of these patients appear to have increased frequencies of T cells and reciprocally decreased frequencies of neutrophils that appear to be associated with good clinical outcome. When we then compare this to patients with severe disease that have eventually recovered, we broadly see a similar response. So the crossed squares here represent severe patients at the time points that they were admitted in ITU. And what we can see is that in these patients as well, over a period of time, there is restored frequencies of T cells and neutrophils that associate with hospital discharge and improved symptoms. However, when we compare these results to severe patients that had a poor outcome, we see that unfortunately in these patients, the frequencies of T cells remain low and the frequencies of neutrophils remain high. Both these patients represented here had critical disease, and the patient represented in orange was in ITU for an extended period of time, whereas the patient in red sadly passed away in hospital. So what we can see from this is that there clearly is an association of restoration of T cell and neutrophil populations with good clinical outcome for disease. Moving on to B cells. So B cells are known to play multiple roles in the immune response against pathogens. So these include the production of antibodies, the presentation of antigens to T cells and NK T cells and their subsequent activation, as well as the production of cytokines. So similar to T cells, B cells can be classified into different subsets based on the types of cytokines they produce, namely B effector cell subsets that produce pro-inflammatory cytokines such as IL-6 and TNF, and regulatory B cells with immunosuppressive capacity that produce anti-inflammatory cytokines such as IL-10. We first measured the frequencies of CD27 high, CD38 high plasmablasts or antibody secreting cells in patients with COVID-19 and found that these were significantly expanded in disease compared to controls. However, we did not find any variability in the frequencies of plasmablasts across disease severities. You will notice here that there are groups of patients across all severities that express have either an expansion of plasmablasts or not. So I just want to make a point that these measures were taken at the time of hospital admission, and a majority of these patients that displayed low frequencies of plasmablasts at this point eventually displayed an expansion that associated with a good antibody response. In addition to plasmablasts, B cells can be categorized into 4 major subsets based on their expression of IgD and CD27. These include naive B cells, unswitched memory B cells, which express IgM, class-switched memory B cells that express either IgG, IgA, or IgE, and double negative memory B cells or atypical memory B cells that are expanded with age and in patients with chronic inflammatory disorders. When we compared the frequencies of these B cell subsets in patients compared to healthy controls, we found that there was a dramatic expansion of the double negative memory B cells in patients with severe disease compared to those with mild and moderate disease, as well as controls. Looking at the other immune cell subsets, we found that there was a general reduction in frequencies of unswitched memory B cells, but no major differences were observed in switched memory B cells, as well as total naive B cells. Within the population of naive B cells, there also exists a population of CD24 high, CD38 high transitional B cell subsets that are known to be the precursors of immunosuppressive regulatory B cells in humans. So these transitional B cells, upon activation via CD40 or toll-like receptor 9, can differentiate into IL-10 secreting immunosuppressive cells that play an important role in suppressing inflammation in health and disease. In patients with severe COVID-19, we can see that there appears to be a decrease in the frequency of these transitional B cells compared to patients with mild and moderate disease, and that in contrast, mature B cells appear to be expanded in majority of patients with disease. When we stimulate PBMCs with CpG, so a TLR9 agonist, or R848, TLR7 agonist, we see that there is a significant reduction in the production of IL-10 and a significant expansion in the production of IL-6 by B cells from patients with COVID-19 compared to healthy controls. Our preliminary findings do not suggest any significant differences in TNF-alpha production by B cells. So overall, this data suggests that there may be a shift in the B cell population towards more effective pro-inflammatory cell subsets with a reduced frequency of immunosuppressive regulatory B cells in circulation. So to summarize what I've shown you so far, the restoration of T cell and neutrophil populations is associated with a good clinical outcome. And we think that using neutrophil to T cell ratio could be used as a biomarker to predict eventual severity of COVID-19. Within the B cell subset, we see a dramatic expansion of the double negative memory B cells in patients with severe disease, mirrored by a slight decrease in transitional B cell frequencies in these patients. Overall, it appears that the B cell populations in patients with COVID-19 appear to be skewing towards a more effective B cell population that actively secrete IL-6, with a reduced frequency of IL-10 producing B cells in circulation. And this is likely one of the phenomena that contributes to the ongoing hyperinflammation and pathogenesis of disease. So I'll stop with that and hand over to Lizzie, who's going to share some exciting monocyte data in COVID-19.
Okay. So there's been several studies, including ours, that have looked at adaptive immunity in COVID-19. It's given a lot of really informative information, but we're interested also in what happens to the innate immune system, and in particular with monocytes and neutrophils. We know that under inflammatory conditions, tissues send out these long reads, including cytokines. This then goes to a different in the bone marrow, and including cells such as monocytes and neutrophils. So these then leave the bone marrow, they enter the blood, and they migrate back to peripheral tissues to carry out their effector function. So we know that monocytes play a critical role in respiratory infections. So for example, during infection in the lung, monocytes are mobilized from the bone marrow. They come out of the bone marrow expressing CCR2, and they're recruited into the lung. And in the lung, these monocytes have pro-inflammatory functions, they're antiviral, they produce a lot of inflammatory cytokines. But it's this production of inflammatory cytokines that means that if this isn't regulated properly, this contributes to excess inflammation, tissue damage, and fibrosis. So we know that from previous studies, monocytes are dysregulated in COVID-19. So this is an X-ray showing a patient with severe COVID-19 pneumonia, and you can see this white populations here are infiltrates containing monocytes and monocyte-derived macrophages. We know that the bronchoalveolar lavage fluid from the lungs of these patients has an abundance of monocytes in. They have a pro-fibrotic signature, and there's an abundance of chemokines that will attract monocytes in, including CCL2 and CCL17. So we know that from post-mortem tissue, these tissues are packed full of monocytes from COVID-19 patients that have deceased, and this isn't just restricted to the lungs, this is also the heart, the kidney, the liver, and the muscles. So these recruited monocytes during infections are inflammatory, and they've got these pro-inflammatory properties that help to clear viral infections. But if this isn't regulated properly, these cytokines can contribute to cytokine storms and tissue damage. And similarly, these recruited monocytes contribute to tissue repair. But if these pathways aren't regulated properly, this can contribute to fibrosis. So we wanted to know in COVID-19, how are circulating monocytes altered? Can these monocyte changes in COVID-19 tell us who's going to go on to have severe disease? So who's going to go on to require intensive care admission? And can we tell this early on at hospital admission? We want to know what happens to the monocytes during COVID-19 disease trajectory. So when should we target these patients for therapeutics that are going to target the immune system? So in normal healthy individuals, monocytes express the cell cycle marker Ki67 in the bone marrow, but monocytes don't express Ki67 in the blood, so they lose expression of Ki67 before they exit the bone marrow into the blood. But what we found that was in COVID-19 patients, circulating monocytes did express the cell cycle marker Ki67. And furthermore, this was at its highest level on patients with severe COVID-19 disease that required intensive care admission. We found that accordingly, levels of Ki67 tracked with C-reactive protein, a clinical indicator of inflammation, and these bloods were taken at the earliest time point after admissions. This is early in the disease course. So the most likely phenomenon for this is enhanced release of monocytes from the bone marrow, so whilst they're still retaining their Ki67 expression. And this is a process known as emergency myelopoiesis, and it's something that we've seen before in H1N1 pandemic influenza. So equally remarkable was the reduced expression of COX-2 in circulating monocytes from COVID-19 patients. So cyclooxygenase 2 is an enzyme that converts prostaglandins from the oxidation of arachidonic acid. So prostaglandins are pro-inflammatory mediators. They accumulate at sites of inflammation and they have antiviral functions. So because viruses have adapted mechanisms to escape host-immune defenses, for example, hepatitis B blocks COX-2, reducing levels of prostaglandins, and this supports viral replication. But of interest as well, non-steroidal anti-inflammatories, including aspirin and ibuprofen, also act on the COX-2 pathway, and they exert their anti-inflammatory function by reducing levels of prostaglandins. So we found that in COVID-19, patients express lower levels of COX-2 on their circulating blood monocytes. And again, this tracks with disease severity. So we think that Ki67 and COX-2 may be early indicators of disease severity that could be used upon admission to stratify patients for treatment before they've gone on to become severe and before they've gone into intensive care. So we wanted to know what happens to these monocytes throughout the disease course. And these experiments, we followed up the same patients, so mild patients who obviously recovered a bit quicker and were discharged, so we have fewer time points from these, and severe patients who we had longer time points from, some of which eventually recovered, but all of which ended up in intensive care. So we've got the mild patients here in green and the severe patients here in black. And we found that with Ki67, the cell cycle marker, this was high in all patients, higher in the severe patients early, so upon admission before ITU, but regardless of the disease outcome, Ki67 levels dropped. So this is more of an early severity predictor. And even in the patients that went on to intensive care and didn't recover, the Ki67 levels still normalized back down to this dotted line here, which is the healthy control levels of Ki67. So there's not very much in the blood of healthy controls and monocytes. But what we found with COX-2 was COX-2 levels increased in the mild patients who recovered. So upon recovery, COX-2 levels came back up to this healthy control level here. But in the severe patients who are in intensive care, the COX-2 levels stayed low. So we wanted to know, as well as the monocytes, what happened to cytokines in the systemic circulation throughout the COVID-19 disease course. The major pro-inflammatory cytokines that we saw increased in the circulation in COVID-19 patients were IL-6, IP-10, and MCP-1. So these further support an important role for myeloid cells in COVID-19 pathogenesis. And we wanted to see what happens as we go throughout the disease course in these patients. So again, we've got the mild patients here in green, we've got the severe patients here in black. And what we found that we see a systemic cytokine level drop even in the patients that didn't recover. So we know that these are higher in the severe patients. I've shown IP-10 here as an example. So these are higher here in the black circles and triangles. But even though these patients didn't all recover, and went into intensive care, the cytokine levels dropped quite early into their hospital stay. So these were high upon admission. And even this patient here with the triangles who sadly died, the levels of IP10 still went down after they were admitted to ITU. And this was the same for IL-6 and MCP1. So this indicates there may be a critical window for targeting immune responses, which is early after admission, so before these patients have actually got to the point where they require intensive care treatment. So in summary, we think we can use this enhanced Ki67 and reduced COX-2 as an early indicator of disease severity, and this may be used to stratify patients upon admission and to stratify for different treatments and to predict who's going to go on and become severe. So the most likely explanation of the increased Ki67 is this premature release of monocytes from the bone marrow whilst they're still retaining Ki67s, this emergency myelopoiesis as seen in pandemic H1N1 influenza. The reduced levels of COX-2 and monocytes indicates that there may be lower levels of prostaglandins produced. This may well lead to lower levels of being able to control the virus, so enhanced viral replication, and in particular in the lung, which is something we're looking at now. These data also potentially indicate that the use of non-steroidal anti-inflammatories may be undesirable in COVID-19 because this will further reduce the levels of COX-2. So this low COX-2 and high IP10 profile is something that's been seen before in idiopathic pulmonary fibrosis. This is this pro-fibrotic profile, and this has really important implications, especially in the severe patients who are now reported to have long-term problems with lung fibrosis. So our results do have clinical implications. As mentioned, the patients that go on to have severe COVID-19 can be identified upon admission when they're not necessarily in their severe stage yet. And this is indicated by the high Ki67, the low COX-2, and also the low T cells, low B cells, and high neutrophils. We know that the cytokine storms, the pro-inflammatory cytokines that we're seeing that are increased in the circulation, is a relatively early event. So at least early within their hospital stay. It's high when they come in on admission in the severe patients, but this reduces whilst they're after admission into intensive care, indicating there may be this optimal treatment window just after admission. These data also indicate the use of non-steroidal anti-inflammatories may be undesirable. This may reduce COX-2 further. And these data also provide pathways that may be targeted for alternative therapeutic options. For example, targeting pathways involved in recruitment of immune cells, pathways involving bone marrow release of cells, so trying to inhibit the release of these abnormal neutrophils and abnormal monocytes into the circulation, and also pathways that will target neutrophil survival, monocyte survival, and also neutrophil and monocyte function. So what are we doing next? We're still actually currently getting a lot of patients coming in that are in this acute phase. So patients that are admitted to hospital with COVID-19, so we're following up with some of the functional implications of these data. But we're also looking at long-term effects of infection on patients. So these early patients that came in March, we're following up what's happening with them now. What are the effects on patient health? We know that lots of patients are reporting that they're suffering from fatigue, depression, so it's not just lung symptoms, and we're looking at their immune responses to see if there are sustained changes in immune responses in these patients that might be contributing to some of these long-term symptoms. We're maintaining the circo structure, and we're all carrying on working together to investigate what happens next, so what happens in subsequent infections. So not necessarily another round of COVID-19, although that's what we're prepared for, but what happens when they're exposed to other respiratory infections. So that could be a new coronavirus, that could be other types of infections, that could be flu, and especially coming into the winter and coming into the flu season, we want to really try and work out how these patients might be responding to other respiratory infections. So thank you to everyone that's been involved with this study. So this has been a huge amount of work from a huge number of people, and it's really been possible because of everyone pooling together and collaborating. So thank you again to everyone. Thank you all for listening, and we are happy to take any questions.
And Lizzie, that was really a fantastic overview of everything that you measured, and obviously your interpretation of what's going on. So there are lots of questions. The first thing I'm going to say is that I'm afraid there have been some issues with the quality of the sound. And I apologize for that. However, this is being recorded and will be available to all of you afterwards as a recording, just to preface with that. So if I could start, I'm monitoring the Q&A here, and really what I want to start with, perhaps if we could clarify the patients and the stratification. So we've had a few questions really on in your healthy controls that I understand were probably you were donated, so basically healthcare workers and people in the university. Did you actually have any COVID-19 carriers who were asymptomatic? Did you check for that at all? Do you know?
So sorry, there's quite a lot of echo here. We tried to get, obviously it was quite difficult to get control blood during the height of the pandemic.
Yes.
So what we've tried to do, as you said, we started off with using each other's blood. But because we're working very closely and we're on site with the Manchester hospitals, we've then been targeting frontline workers who we know from the hospital and asking them to donate blood. So a few of them have been antibody tested and antigen tested. And at the time they were negative, but we've got quite a few controls that we don't know whether they were asymptomatic carriers. So we're antibody testing them in the next month, but no one's had symptoms. So they were either asymptomatic or haven't had symptomatic COVID-19. But we have, we've been quite careful to try and get a cohort separately that's a slightly older group and also skewed towards males because of the gender bias in the patients. So we've done several things to try and correct.
Yes, yeah, and obviously the project is ongoing and you're still collecting. Yes, yeah. Well, maybe with the reversal of lockdown in the UK, you'll be able to get more controls.
Just to add to that, we didn't see increased antigen-specific antibodies in any of our controls. But one of the things we're aware is that people who have mild symptoms don't tend to have high levels of antibodies in circulation. So we are now looking at memory T cell and B cell responses to confirm whether or not a healthy cohort were in fact asymptomatic carriers.
Okay, yeah, thank you. So I think the next question, quite a few questions then. So you basically found what's been reported by others, and that is this real drop in T cells in the blood and increase in neutrophils. So so a couple of questions, is that related, I think it is, but related to absolute cell numbers as opposed to frequencies? So was there not simply the percentage of T cells was going down, the percentage of neutrophils going up, but the absolute numbers? Can you answer that?
Yes, so that was the case. It wasn't just the frequency, it was definitely the absolute numbers of the cell types as well.
Yeah. And you mentioned, I think, Lizzie, you started off mentioning what's been found in biopsies or post-mortem analysis of a high monocyte level. Is how much do we know about other immune cells that are actually infiltrating lungs and other organs? How much do we know about that?
Yes, I think from what I've read, then it's not just monocytes, there's T cells that are infiltrating as well, which is interesting in terms of the depletion from the blood, whether some of that is at least accounted for because maybe they're going to the lung. As well as perhaps there are just fewer T cells. So what we're trying to do now is get hold of, we have actually got hold of some of the bowel from these patients, but we're just getting the procedures in place to do the Cat3 facilities to look at this. So this isn't from our own data, this was from what's been reported, but it seems to be there's certainly monocytes and T cells at higher levels than non-COVID patients in bowel. In terms of other tissue, non-lung tissues, as far as I know, it's mainly monocyte infiltration, but the lung itself, there is more of a mixed population, as you say, so the T cells are increased as well.
Okay, thank you. So the next question, I think one was asked about the antibody response. So how does the antibody response map onto the B cell response? Do you have that data, Madhvi?
Yeah, so we've actually started doing that a couple of weeks ago because we banked the patient's serum and we did the real-time flow cytometry analysis. And we're now looking back at the serum and correlating antigen-specific antibody responses with that. So what we have found is that typically we see increased levels of antigen-specific antibodies in patients with severe disease compared to those with mild and moderate disease. So one of the explanations for this could be that maybe the patients at the time of hospital admissions that have really high levels of antibodies, these are not actually effective at neutralizing the virus and could potentially be contributing to antibody-dependent enhancement and then ongoing inflammation and disease.
Thank you. So then I think a question that everyone wants to know the answer to. So your COX-2, reduced COX-2 levels in the monocytes in the blood, really, really interesting. So I have a specific question. I'm just going to jump in with a question. Is that a direct reflection of reduced COX-2 on monocytes as they're developing in the bone marrow? So is it that monocytes coming out of the bone marrow are low COX-2 as they are high Ki67?
Yeah, so most of the data is actually shown after stimulation, but I have done some experiments now with just fresh on blood that's nothing's been done to it, it's just fresh staining, and the COX-2 is still lower in the COVID-19 patients. But what's interesting is, and one thing we're following up now, is the kinetics look very interesting. So depending on how long you stimulate for, so these were only 3-hour stims with LPS, it's still lower, but then it does start to change and go back and forth quite a lot. So when we stimulate for longer, then the COX-2 actually starts to come up in the patients. So it might be this kind of COX-2 prostaglandin feedback that's then skewed in the patients that's different. So it's not necessarily going to mean that there is going to be lower levels of prostaglandin, I think, in the lung. So we need to follow this up with the kinetics, I think. But certainly coming out fresh, there's still lower COX-2.
So what do you think the benefits of dexamethasone are then?
Well, I guess it's general suppression of, and it's early because it's given as soon as patients come in, right? So it would have a general immunosuppressive effect, which would fit with what we're seeing in that we see all the IP10, the IL-6 and the MCP1 early before patients have gone into intensive care. So in terms of general suppression, then that's going to be useful, but there's quite a few patients that it's not working on. So we're trying to kind of stratify down a bit more to see, even within all of the severe group, they still look quite different, and there's going to be differences depending on what their comorbidities are. Some are super high for IL-6, for example. We've actually had 1 or 2 where their IL-6 stays high during ITU, even though most people's does drop off. So I think once we can start to pull apart what the differences is there, we can maybe target a bit more specifically to try and help the ones that aren't responding to dexamethasone.
Yeah, thank you. So Madhvi, a question for you about the B cell responses. There's a lot of interest in the double negative B cells and increasing plasma blasts. Can you say a little bit more about that, what you think is going on, and maybe what it's everyone, who's, because obviously a lot of centres around the world are, you've shown really beautifully how you've been able to coordinate and come together very quickly to respond to this pandemic and any future pandemic, as you say, or virus infection, which is fantastic. So are other people measuring B cell markers in the same way that you are? And
yes, so this expansion of double negative B cells that we see has also been reported by another group, by Ignacio Sanz. They also find a similar expansion of double negative B cells in critically ill COVID patients compared to those with mild and moderate disease. And double negative B cells are quite a heterogeneous population or subset of B cells. So they're known to contain subsets of extrafollicular B cells that, for instance, in chronic inflammatory diseases like lupus, they rapidly respond to toll-like receptor 7 activation and produce large amounts of IL-6 and secrete antibodies as well. So one of the explanations is that this expansion of double negative cells that promotes a more pro-inflammatory response, combined with the reduced transitional cells and reduced IL-10, could suggest a shift in the balance of B cells in COVID-19 patients. And within the double negative cells, they are known to play an important role in antiviral infections. However, in one particular study looking at HIV patients, they found these cells to be associated with senescence and more of an exhausted phenotype, as well as associated with T cell exhaustion. So these cells expressing PD-L1 were found to contribute, were speculated or rather associated with T cell exhaustion.
And then a few questions relating to the patient treatment. What sort of therapies did they go on, as it were, in Manchester? What do you do in Manchester? What's done in Manchester hospitals?
I guess it's sort of changing as we go. So certainly initially the major treatment was sort of respiratory support. So they weren't necessarily, if they go into intensive care, most are on antibiotics to try and prevent secondary infections. But we've really, really gone through in depth all of the clinical data because obviously, especially with the COX-2, where are they all on non-steroidals, for example, and is that why their COX-2 is low? But they're not, so some are, some aren't. We tried to take that into account. To start with, it was just general respiratory support and antibiotics, but obviously as time's gone on, there's more and more trials that are going ahead. So there's the IL-1 receptor trial that's going on in Manchester. Some of the patients are on that, not for our study, but there's now obviously patients on dexamethasone as well. So various different types of immune modulatory therapies are starting to come in. But there's nothing apart from sedation in terms of when they go into intensive care initially. There wasn't some one drug that seemed to be causing this, unless it's the sort of sedation itself in the severe patients.
And how long would the patients be followed up for? I guess an interesting thing, follow-up, is going to be those that were in ICU and recovered, whether they have obviously permanent lung damage or the chronic fatigue syndrome that's associated with the virus infection, post-viral syndrome. Yeah. How long will they be followed up for?
So
conducting a follow-up study where we're getting patients 12 weeks post from the start of symptoms to come back and link their fatigue or any other symptoms that they display with their immune response. And Lizzie actually has some really interesting data on monocytes at the moment that maybe you want to talk a little bit about.
Yes, I mean, it's pretty early, haven't we, this week? But it does look like there's some level of longer-term dysfunction still. So in these follow-up patients, in which we're looking into exactly what it is and how they respond to different types of stimulation and things. But certainly, these are 12-week follow-up, which actually we were surprised when these bloods came in. We didn't realise it's been 12 weeks since they've been in. It's just the time's been really quick. But
it's gone really quickly. Yeah, it's been a very intense time. Yeah.
But they do seem to have these quite long-term changes in their monocytes, and we're very, very interested in the chronic fatigue, as you said, because lots of the patients are reporting, even the patients with milder infections are reporting these chronic fatigue-like syndromes. That's very, very debilitating, I think. So we're really interested in if this tracks with some of the monocytes that have got certain profiles and something that now on some of the patients.
So there were a few questions relating to other potential biomarkers. So cytokines. So you highlighted IL-6, IP-10, and MCP-1. So a few questions related to things like IL-12 or IL-2 or IL-10. Did you monitor all of those and you sort of eliminated them as stratifiers for the different categories of infection or severity of disease? Or?
Yes, we included a whole range. I think it was 37 cytokines within the Legendplex kit. We did actually see an increase in IL-10, which I should have mentioned before, but we've so IL-10 is increased in the patients as well, but we didn't actually see any differences in IL-12. We didn't see any differences in TNF, which was surprising. There was a bit of a trend with TNF, but we didn't actually see this increase that some of these cohorts in America have been quite striking. And so that's also something we're wondering if that's to do with just differences in patient demographics. We're looking through with collaborators in the States for the differences between our cohorts, whether that's to do with age, BMI, ethnicity. So we're trying to talk through everything with statisticians now to see what's the differences between these cohorts. But really, the only other cytokine that came up in the legend paper was IL-10.
Right, okay. And interferons, so type 1 interferons, what do they look like?
So we have some very, very preliminary data as of last week that suggests that there is an impaired type 1 interferon response in patients with severe disease compared to those with mild and moderate symptoms. And this is similar to what other people have found and reported in the literature as well. And I think the idea is that sort of links up with the delayed antibody response as well in some individuals, because type 1 interferons play a role. So interferon alpha and IL-6 together are important in differentiation of plasma cells, antibody produced secreting cells, and that there could be some link between type 1 interferons and impaired B cell response.
Okay, interesting. So I'm just going to have a quick look at the other. But while I'm doing that, I'm going to be really selfish and ask you, because you are setting up your own laboratories. So you're I should have said that Madhvi is a presidential fellow that's just been appointed, and Lizzie is a Henry Dale research fellow. So just tell us a little bit about your experiences of this responding to the pandemic, and the sorts of, if you can remember, before we had all that stress of lockdown, the thought processes, because that is, I think that would be really informative. How you came together in the collaboration, and what it was like.
For me, it was really interesting because I just arrived at Manchester and then everything shut down and I didn't know anyone and I wasn't sure what exactly I was doing. We have this, so every Friday we have these meetings, immunology meetings, where people come together and we discuss things that we can do together and collaborate within the division. And Tracy Russell, who is the director, really took the lead in setting up a team that worked together and brought together our own bits of immunology expertise to work on addressing these questions. And I'm trying to think back when we first started. For me personally, it was a great experience because I got to work closely with Lizzie and John, Joe, Sean, and Tracy for the project. And I think in addition to a great work, it's been also close friendships that have formed. So it's been really fantastic working together.
Mm-hmm. And Lizzie?
I just reminded me to say, because these Friday 4:00, they're called Bill Ford Fridays. Or BFFs, we call them best friends forever.
Yeah.
So we had Tracy mention that there was an opportunity to get this project together and who wanted to be involved, and we all just kind of came together and it just sort of worked really. So we spent the first couple of weeks thinking about what we wanted to do and ordering and panels and what would be the easiest, who wants to do what, and it just sort of came together really. But it was great to get to know Madhvi better because we ended up doing quite a lot of the lab work early on together, but it just made it a lot easier when there's two of you doing it together. And then we've had lots and lots of help since then, as we've had so many great PhDs and postdocs that have come in to help with all of the analysis and the processing, and it just expanded out in the end to enable us to get quite a lot of data. But it's just sort of happened each week. Just I can't really explain more and more. It just sort of worked.
Yeah. So I'm going to then be a personal touch. So I actually started my research career with Bill Ford in Manchester.
Yeah, that's what I mentioned.
Immunology, yeah. So this is where I started on T-cell trafficking and high endothelial venules. So Bill actually would be so proud that there's a Bill Ford Friday. I love it. I absolutely love it because that's exactly his attitude, was to get everyone together and exchange ideas and see what could be done together on a collaborative basis. So that is very heartwarming. That's very heartwarming. So for me personally, but that's been very selfish. So then really, we won't, I think, just perhaps tie up now. I think we've covered most of the questions. A sort of $64,000 question really for the monocytes is because we know, we have so much information now about patrolling monocytes, classical monocytes, intermediate monocytes, non-classical monocytes. Do you want to say anything about that, Sir Lizzie? Or
yeah, so all the data I showed is just CD14 classical monocytes, but we have done a lot of work subsetting and looking at what's going on with the different subsets. And similar to other studies, we are seeing an expansion of the intermediate CD14, CD16s, and they're very, very pro-inflammatory, both in the controls and the patients. But they are expanded in the patients. So we think there is a role for these, which we're now looking into further, because we've just sort of put them to the side for now, just in terms of time and getting the kind of major messages out. But in terms of what they're doing, if they're going into the tissue or not, if they're then contributing, because they're really, really potent TNF producers, they're potent IL-1 beta producers. And actually we see a drop of IL-1 beta from the classical monocytes, but these are producing a lot more in the patients. And we also see an absolute loss of the non-classical CD16s. They're just completely gone from the blood. So this is why we really want to start with the BAL and see what it looks like. Are they just actually not coming out? Are they not there at all? Or are they going into the tissue? But we're very something that ties in quite nicely with what I was working on before, is how this is going to affect tissues longer term. So I'm looking at monocyte infiltration into tissues and development into macrophages. Was already looking at that in the intestine and also in the reproductive tract. So I'm really interested to see what are the kind of longer term effects in tissues like the intestine and also the reproductive tract that have got a really, really high level of replenishment of macrophages from blood monocytes. So we're seeing the uterus is even more replenished than the gut. So these are the tissues that we think are probably going to be affected long term if we are seeing these long-term dysregulation.
So this is applying what you've learned from COVID-19 to your fellowship, or what your first love, as it were. You know what I mean? And fantastic. Yeah, and I guess for you, Madhvi, as well. You will use the information here, what you've learned, for your fellowship, and what you want to do. It's fantastic. Fantastic. So I think we have probably exhausted both of you, and I think we've covered most of the topics on the Q&A session. I just really want to thank both of you. It's really, really wonderful to hear from you, because we've seen you. I don't know how international it is, but I've seen you on the Channel 4 news, and I've seen you in the newspapers working together, full PPE in the Class 2 hood, handling infected blood. So now we're seeing the results of it. So this is fantastic. So I'd just like to finish by really thanking you on behalf of IUIS and Frontiers, this collaboration. And really invite you to really respond to email queries from the audience whose questions maybe we haven't been able to address in the way that they would like in the time we have available. Okay, so if I could clap, I shall clap you. Thank you very much indeed. And thank you to everyone around the world for listening. And as I said, there will be a recording available for this fantastic webinar. Thank you so much. Bye-bye, everyone.
Thank you.
Bye. Bye. Bye.